last section, 1-3 of Chapter 1, which is going to be the methods of science. Basically, this is just a review of the scientific method. And you had this last year in 9th grade or possibly even 8th grade. So, and if you take science next year, you're going to have it again. Every year we review the scientific method. So here we go. So the first step to the scientific method is to ask a question. Science always starts with an observation, not with an S, with an observation. What is an observation? An observation is a direct method of gathering information in an orderly way. So, what do you think we use to make these observations? Well, we use our senses, and that would be sight, hearing, touch, smell, and taste. So what exactly is the scientific method, or those steps? An inference is a logical interpretation based on prior knowledge. The scientific method is a similar method all scientists use to gather data and answer questions. And really, you're using the scientific method most of the time when you're making decisions without even realizing that you're using the scientific method to come to a conclusion. So, I've got a chart on the next slide, and we'll see if you can guess which ones are observations and which ones are inferences. So here's our chart. Object A is round and orange. Would this be an observation or an inference? This is an observation. Object A is a basketball. That is an inference. Remember, an inference is a logical interpretation based on prior knowledge. How about the next one? Object C is round and black. Perfect. Observation. Object C is larger than B. Once again, observation. Next one. Object B is smooth. Observation. And of course, if you look over here, these are the objects that we're talking about. Object A is a basketball. Object B is a table tennis ball, which is a ping pong ball. Object C is a soccer ball. Next one, object B is a table tennis ball. That is an inference. And the last one, kind of tricky, each object is used in a sport. That is actually both. And there you have it. So, after we do the ask a question, make an observation, we then have to form a hypothesis. And a hypothesis is a testable explanation of a situation. Sometimes you hear it referred to as an educated guess, but that's really not the scientific definition of a hypothesis. A hypothesis is only useful if it can be tested through a controlled experiment. And this is the key to it all, a controlled experiment. And we're going to go through what a controlled experiment is. When a hypothesis is supported by data from multiple investigations, another key right here, multiple investigations, the hypothesis is usually considered valid. Valid means true, and accepted by the scientific community. So once again, a hypothesis is supported by data from multiple investigations. That's the key. So once we form a hypothesis, then we need to collect our data by doing an experiment. And an experiment is an investigation in a controlled setting to test a hypothesis. So once again, here we've got that word, control. And what does that mean to have a controlled experiment? The control group is an experiment in which the group is used as a comparison. That's a really bad sentence there. So the control is what we're going to compare the experimental group to. So this one, the control, always stays the same. The control always stays the same. We never change the control. The experimental group is a group exposed to the factor being tested. So the experimental group is the one that we are testing. So, for example, if we were doing an experiment on these two plants, we might add water to this one to see if it's going to grow. This one, we're not going to add water to because it is our control. We're changing the variable here by adding water. So if we add water, then the plant will grow. Oh, there's your hypothesis also. So experimental design. In an experimental design, once again, there's going to be just one. And here's the key, because I'm going to ask you this on the test, and we're going to ask you this all along. Only one factor can be changed at a time. We can't change more than one, because if we change more than one, we don't know which factor is affecting what we're testing. So we only change one, just one. So the control, or sometimes called constant, is a factor that remains the same. Once again, remains the same during an experiment, while here's what changes: the independent and dependent variables change. So the control, once again, this always stays the same. These two are what we're going to change. So, what is the difference between the independent and the dependent variable? The independent variable is the tested factor that might affect the outcome of the experiment. So once again, the independent variable is the tested factor that might affect the outcome of the experiment. The dependent variable results from or depends on changes to the independent variable. So once again, the dependent variable is the result from or depends on the change to the independent variable. So in this particular experiment, the independent variable is the light. The dependent variable is going to be the growth. So once again, the light is the independent variable. The growth, or what we're going to see happen, is the dependent variable. And this is just another slide once again, explaining. So the independent variable is what I change. The dependent variable is what I observe. And then the control is what I keep the same. Okay, so we then experiment, and the next thing to do is we need to collect data. And data is information gathered from our observation or from our experiment. And there are two types of data. There is quantitative, which are numbers, and in this quantitative, you can see the word quantity. So, an example of that would be, there are 7 birds at the feeder. That is quantitative, 7 birds at the feeder, data. The other type of data is qualitative. Notice there's an L in there. This is a descriptive type of data where we're using characteristics that cannot be counted. So an example of qualitative data is the bird has a red head. That's describing the bird. Okay? The metric system is what scientists use in order to count their data. And we use units with divisions that are based on powers of 10. SI, or the International System of Units, are commonly used in science for consistency and ease of communication. So, SI is the same thing as the metric system. This is our common term that we use in order to talk about the system that we're using for measurement. SI is what they use in the science world. Okay, so once we have collected our data, the next thing that we need to do is analyze our data. So after data collection, scientists use analysis to ask questions such as, has my hypotheses been supported? And what scientists look for are patterns in the data that can be displayed in tables, charts, or graphs. And as we know, this is what you just completed that lab on. I asked you to draw a table, and from the data in the table, I asked you to make a graph, and you also made a pie chart, and you also made a bar graph. So you, in the lab, demonstrated different ways in order to analyze data. And then finally, once we analyze that data, we form a conclusion. And before a scientist can publish their findings, it must be peer-reviewed. And once again, peers are their colleagues or the people that are in the same field that they are working in. For example, your peers are going to be the rest of the students in your class or in your high school. Those are your peers. Something that you share your life with, they're around the same age, those are your peers. If the reviewers agree, it is then published in a scientific journal. Others can then use your data to evaluate and develop their own research. So by publishing their findings, other people can then go further in researching whatever they are working on. And finally, student scientific inquiry. Before starting any activity, read all of the steps and make sure that you understand the entire procedure, including any safety procedures. This is one of the most important things, and if you come and ask me questions in lab, I will make sure to ask you, did you read the directions carefully? Always follow my directions. Ask for an explanation when you are in doubt about any part of the activity. If you don't understand what you're doing, please ask. And finally, because we do not know what goes on in lab, wash your hands thoroughly after every scientific activity. That's extremely important because we don't know who's been in the lab, who's touched the equipment, and so forth. So that concludes Chapter 1.